withdrawal and negative.
There she is. So Dr. Valentina Vozella, she's now at the Scripps Institute in California. Valentina actually was a PhD student in IIT, my institution. She also spent some time in my lab, and while she was there, I realized that she had a brilliant future ahead, and this is exactly what happened. And so I'm really happy to have Valentina here today. She's a young, brilliant Italian scientist that is trying to make the difference abroad and in one of the most important research institutes worldwide. So please, Valentina, start your presentation and let us know what you're working on.
Can you see my presentation?
Yes. You can start, Valentina.
Yeah, I don't know why it's
In
not
presentation
going
mode, please.
Okay.
Good.
Can you see?
Right.
Perfect. All right. So thank you so much, Andrea, for the really kind and nice introduction and for inviting me for this IMaSS Lipidomic Day today. So as Andrea was mentioning, I'm now working at Scripps Research in San Diego, California. And today I'm going to present part of the work that I've been doing as a postdoc here. I'm going to switch gear a little bit compared to the previous few presentations and show you how we employ mass spectrometry and lipidomics to address biological and pharmacological questions in the field of alcohol use disorder and stress. So in my lab, we use in vivo pharmacological, behavioral, and lipidomical approaches to study the endocannabinoid system and the relation with stress and alcohol use disorder. And our ultimate goal is to accelerate the discovery and development of new therapeutic options. Some of you in the audience might be already familiar with the endocannabinoid system, which is represented here on the left side. As you can see here, the endocannabinoid machinery is pretty complex. There are receptors called endocannabinoid receptors, lipid mediators such as N-arachidonyl ethanolamine, also called anandamide, and 2-arachidonyl glycerol, or 2-AG. And the enzymes involved in these lipids synthesis and degradation, such as monoacylglycerol lipase for the degradation of 2-AG, and fatty acid amide hydrolase for the degradation of anandamide and several other lipids. What makes endocannabinoids different from other neurotransmitters is that they are lipid derived, and they are synthesized from membrane phospholipids. They are not stored in cell vesicles, but they are synthesized and released after and immediately released. And after they act, they are immediately degraded. The endocannabinoid system affects a large number of physiological and pathological processes, including learning and memory, neurodegeneration, motivation and reward, stress response and mood regulation, and others. So with this cartoon, I just want to give you the framework for the biological basis of substance use disorder. In the field of substance use disorder, we refer to it as the cycle of addiction and involves three main stages: binge and intoxication, withdrawal and negative affect, negative emotions, and preoccupation and anticipation. These three stages involve dysregulation that are mediated by three main brain regions: basal ganglia, the extended amygdala, and the prefrontal cortex. These three areas are organized in subregions that crosstalk with each other. And the central nucleus of the amygdala mediates fear and anxiety and provide motivational drive for consumption of alcohol. The prelimbic and infralimbic cortex are the two major medial prefrontal cortex areas and are thought to mediate the control of goal-oriented behavior. Amygdala and prefrontal cortex are together part of the corticolimbic system. So to study stress and alcohol use disorder, we use a special strain of selectively bred alcohol-preferring rats called Marquesian Sardinian alcohol-preferring rats. They are characterized by high alcohol preference and high alcohol-seeking behavior, increased anxiety, and decreased anandamide in the extracellular space of the central amygdala. The family of lipid neuromodulators has been growing a lot in the past few years, since the use of different omics techniques has led to the discovery of a large number of other endogenous N-acyl ethanolamine, other than anandamide, for example, or other monoacyl glycerols that altogether belong to the complex lipid signaling system that is now have been named endocannabinoidome. For example, a study published last year showed that the inhibition of NA, which is here in red in the cartoon, which is responsible for the metabolization of palmitoyl ethanolamide and oleoyl ethanolamide, has an effect in reducing alcohol intake in this strain of animals, alcohol-preferring rats, as you can see here. So what did we do? We first used a lipidomic approach to address the questions. Is the endocannabinoidome dysregulated in corticolimbic areas of this innate high anxiety rodent model? Which are the most affected brain regions? And does sexual dimorphism play a role? So we used male and female high-stress MSPs rats and whisters. Whisters are our control naive animals, so they don't have any sign of high stress or alcohol preference. We sucked the animals and collected the brains that were immediately frozen. The brains were then sliced at the cryostat and micropunches of the region of interest, so central amygdala and prelimbic and infralimbic cortex, so the subnuclei of the prefrontal cortex were collected. And we then extracted the lipids using a very standard lipid liquid extraction method with chloroform and methanol. And then we analyzed our samples using Agilent 6470 triple quad. So with our method, we were able to quantify a broad spectrum of lipids, such as N-acyl ethanolamine on top, monoacyl glycerols, diacyl glycerols, and some of the free fatty acids. To analyze our data, we used the male Whistler group as a control group. And here I'm highlighting with the blue arrows the most significant and lipids that show a significant change compared to the control group. As you can see here, the palmitoyl and oleoyl ethanolamide were changing in the central amygdala, as well as the 2-AG and the MAG22.6 and MAG18.1 here in blue. So when we look at the same data, but focusing more on the MSP strain, again, we saw a clear effect on of the sex, of the different sex on 2-AG and MAG22.6. We then looked also at the prefrontal cortex, and in the prelimbic cortex, the effect was way less marked than the amygdala. But we still observed some changes in the 2-AG and MAG22.6. finally, the most striking effect that we observed was of the stress phenotype specifically, and of sex as well, were in the infralimbic cortex. As you can see, not only 2AG and MAC22.6 were decreased compared to the control. So you can see that they are like strong red, but also several other deacylglycerols were decreased as well compared to the control group. So from this preliminary lipidomic analysis, we concluded that 2AG is decreased in the central amygdala and in prelimbic areas of our female rats compared to the males. And in the infralimbic cortex, we observed not only a sex, but also a genotype-dependent difference. Additionally, we looked deeper into our data, and we observed that lipid containing the fatty acid 24, so arachidonic acid, were found less abundant in MSP when compared to Whistler. Next, since the 2AG data was the most robust and consistent across all the brain region analyzed, we decided to follow up using in vivo pharmacology and animal behavior to address the question, is pharmacological modulation of the endocannabinoid through monoacylglycerol lipase inhibition able to ameliorate anxiety and irritability-like behaviors that are kind of hallmarks of alcohol use disorders. So we used MGN110 to inhibit the MAG lipase and so increase the levels of the 2AG. And well, we ran a preliminary experiment where we just treat the animals and ran a lipidomic, and we observed that MGN not only increased the 2AG, but also other monoacylglycerol lipase, such as the 22.6, 18.1, and 18.2. So what we did, we treated the males and females, MSPs and Whistler rats with either vehicle or MGN. We choose the 5 milligram kilo as a dose, as it is reported to exert the max inhibition in the rat brain. And then we test the rats using irritability and anxiety tests. So in the irritability test, what we do, we basically challenge the rat with a probe, forcing the interaction between the probe and the animal. And we assess aggressive behaviors, which include biting of the probe, boxing, following, and mounting. Defensive behaviors, including startling, digging, freezing, vocalizing, and attempt to escape. And we score how many times the animal show this sort of behaviors, and we repeat it. We had the animal undergoing 10 trials of the same sort of interaction with the probe. And as you can see here on the top, MGN, so an increase of 2AG level in the MSP male was able to increase sort of a defensive behavior, while in the female, interestingly enough, the MGN was able to reduce aggressive behavior in the MSPs. So we did observe a sex difference in the outcome. Then, as I said, we also ran an anxiety behavior, which is the novelty-induced hypophagia, where the animal face a choice between engaging in feeding and the fear of novelty. So this test uses chocolate pellets as a familiar element for the animal. And we put the animal in a different room, different light, different noise, and different cage. So all these elements represent a novel and unfamiliar environment. So the animals are pre-trained to the chocolate food the day before, and then they undergo testing. And as you can see here, we didn't observe any effect in the males, but again, we did observe an effect in the females. So MJN was able to increase pellet intake, which indicates less anxiety. So the animal is less scared, less afraid to go into the center of the new cage, new environment, and eating the palatable food. So by now, I hope that I convinced you and I show you that sex and genotype differences were observed in the endocannabinoidome. The 2-AG is altered in corticolimbic areas of alcohol-preferring rats, and that MGL MAG lipase inhibitors are useful tools for ameliorating symptoms in a high-stress animal model. So with that, with that, I want to thank my lab, my mentor, Dr. Marisa Roberto, my lab mate, Brian, who helped me a lot with all the behavioral experiments that I've been running. I only show you a few examples. What we have been running a way bigger set of behavioral tests, and Joy for helping us with punching all the brains for the lipidomic analysis. I want to thank the Pravat Lab and my collaborator, Anna, who gave me a huge help with lipidomics, and other collaborators that are involved in this project, Dr. Zorilla, Natividad, and Dr. Chico Cioppo from University of Camerino. And of course, our funding sources, the NIAAA, the National Institute of Alcohol Abuse and Alcoholism. And thank you all for your attention, and I'm happy to take any question you might have.
Thank you very much, Valentina. Very, very interesting. Is there any question for her? No, apparently no. Actually, just out of curiosity, did you try to compare the circulating lipidome of this animal, not just the central nervous system, down in the brain level?
Yeah, well,
wondering if you notice any difference in their lipidome, for example, in the serum or in the plasma.
Yeah, we haven't done that yet. We did collect the blood samples, plasma samples, of course, and yeah, I think one of the next steps would be to try to correlate the brain lipidome with the circulating lipids level, for sure.
Okay, that might be interesting.
Yeah.
Andrea, do you have any question? I've seen that you did not. Second curiosity I have is that, I mean, you showed a lot of statistics, but done analyzing one single lipid at the time. I was wondering if you try any multivariate analysis on this data, because based on the first slide, I think that you've been tracking like 30 lipids or something like that. So I was wondering if you did any multivariate analysis to try and see if there are any.
Yeah, no, we haven't.
Differences.
Yeah, no, we haven't done that yet. But these were like very preliminary data, like collected in a couple of months. So we are still processing. Yeah, for sure.
Okay, I would give it a try, honestly.
Yes. Yeah.
Okay. There's no further question for Valentina. Then thank you very much again.
Thank you.